All right, chapter 16, microbes of the environment. So this chapter obviously goes together with the previous chapter, the microbes in agriculture, and now we're talking about sort of the big picture of environment. So after this chapter, we should be able to identify the essential roles of microbes in the carbon cycle, sulfur cycle, the nitrogen cycle, the phosphorus cycle, the oxygen cycle. Describe the 3 steps in wastewater treatment. Identify the sources of water pollution. Explain the role of water treatment process. Okay, so we can see the Earth as a self-contained entity, and we are. You can see this sort of as its own spaceship, where everything it needs to survive, to thrive, is carried on the planet. Now, one of the problems is that many of the Earth's resources are non-renewable. Some of them are. Um, a vast majority of them are not. Okay. Now, we do have a number of things that are renewable, say the carbon cycle. Okay, carbon is recyclable, but we have to use microbes to be able to do that. So let's talk a little bit about these natural cycles. Okay. So the Earth is a self-contained ecosystem, and um, the ecosystem is a community of all organisms and their non-living components. Um, the easiest way to think about that is anywhere where there's oxygen or water is probably the Earth ecosystem. Now, with all the thousands of different microbes, that can be viewed as sort of a superorganism where there might not be, all the microbes each individually are very, very small organisms, but taken together, the sheer number of them makes them a whole organism. Now, within uh, the ecosystem, we have 3 major habitats. That is air, soil, and water. And all 3 of those are critical to life on Earth. And when we are studying air, water, soil, you're looking basically at a field of environmental microbiology. So the Earth in an ecosystem as a whole, you're going to have producers. So those are things that are making some sort of biomass. Um, a easy way to think about that is trees. Okay. Uh, consumers are things that would eat some of the food that the producers are making. Um, from a microbial sense, when a tree falls down, a mushroom is going to take over and recycle that. Um, from a macroorganism point of view, the nuts of a tree could be something that a squirrel could live off of. So both of those mushrooms and the squirrels would be consumers in that case. We can also have decomposition products. Um, the fungi in the previous example uh, are an example of a decomposer. Um, and that is getting into the biogeochemical cycles. And those are cycles I listed on that first slide. Um, and we'll go through each of those now. So in the carbon cycle, uh, we are mostly getting carbon eventually being turned into CO2 as part of the atmosphere, and then the carbon will be recycled into a form that it can be taken up by plants and be used as primary consumers. Um, there's some that is lost in combustion for fossil fuels or from processing plants. Um, but in general, uh, plants and animals are going to be producing some CO2, and eventually we will get some of that CO2 used again in photosynthesis to make plant material. Sulfur cycle, and sulfur being among the most limited of the resources, um, we're basically going to be able to turn um, sulfur into an H2S form, which is a very, very, very smelly gas. Um, and then through microbial action, we can convert that H2S into pure sulfur, and then that sulfur can be produced into a SO4 uh, form, and that SO4 form can be taken up by plants and animals. The nitrogen cycle, which we talked about some in the previous chapter, um, again, the atmosphere is very, very high in nitrogen, but it's in the gaseous form. So it's in the N2 form. And plants and animals can't uptake the N2 form. So that has to be converted into something that can be used by plants and animals, and that is, in general, um, a bacterial process. We can have nitrifying bacteria and denitrifying bacteria, which will convert uh, the N2 into an ammonia form, which can be taken up by plants and animals, and then eventually converted into an NO3 form, and the denitrifying bacteria can turn that back into um, the N2 form. Phosphorus cycle. Um, and when you're thinking about phosphorus, uh, I would remind you that your DNA has lots of phosphorus in it. So you can't have uh, genes being passed on if you don't have a lot of phosphorus around. Um, so it is usually um, sloughed off into water um, and deposited into sediments. But then when a plant takes up some of that water, then that phosphorus can get um, reabsorbed into an animal. And then when we eat the animal, we can get some raw phosphorus. Um, So again, a big cycle, we reuse these things. Um, okay, the oxygen cycle. Well, 50% of the oxygen we breathe is of microbial origin. Um, and so we generally think of plants as being our big source of oxygen, but really the microbes are an even bigger source. Um, Now, where we mostly get the CO2 form is during photosynthesis when you break down uh, glucose, then that oxygen will then get reused during aerobic respiration. Okay, preserving the environment. Um, so, uh, in general, we have to have sanitary and wastewater facilities. Um, at the turn of the 20th century, um, there was a great sanitary movement where there was a big emphasis on cleanliness, and it was shown pretty clearly that if you could uh, recycle the water and get rid of the water in a environmentally conscious way, you could reduce a lot of risks by getting rid of the sanitation problems. Um, Before toilets went directly into a wastewater treatment plant, very often that waste was just thrown out the window. Um, so among ways which the sanitation can be cleaned up are cesspools, septic tanks, and oxidation lagoons. A cesspool is just basically a place to hold all the waste. A septic tank is a tank divided, divided so that the microbes that can break down the waste are going to be present, and we now currently have large uh, oxidation lagoons, which are used to clean that sanitation waste. Okay, so the basic steps in waste ma management is you're going to get your raw sewage from homes, okay, and those will be screened, and a lot of the debris will be removed. Then you have a primary sludge uh, tank where the starting of the anaerobic processes are going to happen. Then you have an active sludge or microbial waste where the water from the primary tank is going to be pulled off, and then that goes through a secondary treatment. After that secondary treatment, the water again is pulled off, and that is disinfectant and usually chlorinated or oxidized, but most often chlorinated, so that the water going back into a stream or body of water is fairly clean. Okay. Um, Now you'll see from the primary and secondary fermentation, we now get that anaerobic sludge tank, and that is where a large amount of that solid waste is degraded by anaerobic processes, producing lots and lots of methane gas. Okay. Biofilms are protected structures where aquatic microbes are going to live. Um, One place that you can see a lot of biofilms is on your teeth. Okay, and that's actually why you have to brush your teeth so often, is because you need to remove all of the biofilms um, that are holding the bacteria onto your tooth surface. Okay. And like it says, biofilms are mobilized communities of microbes, um, and they have this sort of mesh of polysaccharide fibers that keep them attached to aquatic surfaces. Okay. Um, so we can actually use bioremediation, um, and bioremediation is simply the reuse or breakdown of an environmental waste with the microbes. Very often we do these in biofilms, um, and so then the waste is broken down, but the film is essentially sort of permanent, um, and so you don't get rid of the microbes. You can just flush the waste products out of the out of the biofilm. Um, Biostimulation is where you're going to feed something to the microbes so that they do a lot of the bioremediation. Now, where I've had a lot of work with biostimulation is we found that we could, if we fed the biofilms beer, we could get them to do a lot of cleanup of acid mine drainage. Um, and so uh, we obviously don't use good beer. We use waste beer that is pe- people who make beer. They generally waste a bunch of the product before it gets processed, or even after it's been on the shelf a certain amount of time. It has to come back to the plant to be disposed of, and we can use that as a biostimulant. Okay. Bioaugmentation, that would be adding nutrients to help uh, stimulate the natural communities that are there, or sometimes you're adding certain communities to the natural communities so that they'll do more bioremediation. Okay. Um, I'm not as big a fan of bioaugmentation because you never know that the bacteria that you're adding are quite as safe. So in biostimulation, you can assume that there are no new bacteria being added, and so they should be safe. But in bioaugmentation, since you're adding specific bacteria to speed up those processes, you do run the risk of having uh, some pathogens being introduced. Water pollution and purification. Um, so potable water means that it's water that you can drink. And there are many sources of water pollution. Um, we can have industrial uh, causes, or you could have individuals who are adding things to the water. Um, but the point is that whenever you are putting something not natural back into the environment, that is seen as a water pollution. A big problem for things like nuclear plants is thermal pollution. You can get a lot of pollution of the water because they get so hot that it increases the amount of algae growing. Um, I do want to point out if the source of the water pollution is human, um, since humans carry a number of human pathogens in their intestines, you can get a lot of pathogenic organisms into the system, uh, not necessarily intentionally, but just as a byproduct. Now, when we see those uh, uh, when we think we have water pollution, we can look for indicator organisms. And E. coli actually turns out to be one of these indicator organisms. And we use those because fecal material from humans does have a lot of E. coli in them, and it generally not found in just sort of naturally in the environment. Um, and so we have used a number of systems to detect coliform bacteria. Coliform bacteria includes E. coli, things like Salmonella and Shigella, and those are all indicators that there's some fecal contamination in your water. Okay. Now we can assay this with a membrane filter technique where we take a sample of the water and we filter it onto a membrane and then transfer the membrane to a media that will turn the different kinds of bacteria different colors. Um, so the E. coli is generally uh, turn blue on these techniques, and just by counting the number of E. coli present, you can see how much fecal contamination there is. Okay. And so there's a filter with several colonies growing on it, and that colored plate has some sort of dye in it that will only turn a certain kind of bacteria a certain color. So when you're doing water treatment, you start with the reservoir, okay? Then the water is mixed in um, with generally alum. Um, okay. Um, then you can make these large flocks in the water, and the flocks can be removed. Um, and the settling tanks are going to contain all the flocks, and then only pure water will get through. That can then get filtered and then chlorinated and put into storage tanks. And there again, 3 steps: sedimentation, filtration, and chlorination. Okay, so what we should have learned from this lecture: the role of micro in biogeochemical cycles, steps involved in waste treatment, use of microbes in bioremediation, biostimulation, bioaugmentation, direct and indirect sources of water pollution, and methods for detecting pathogen polluted waters. And finally, steps in water purification. Um, and again, just a thought question is, how are biofilms used in all the different topics that we see in this chapter?